So I'm going to do a review as fast as I can for your final exam. The details of the contents are all in previous lectures. So to save time, because there's so much material, I'm not going to go into a lot of detail about anything, but I just want to give you the overall picture of what material is on the final exam, which you should know from study guide and the quizzes and everything that we've just kind of been through. I'm going to just give you this information right now. So what we have are really kind of the whole majority of the material, which is 100 points of material on the exam, is going to be really on genetics, but we kind of have two aspects of this. We have chromosomal, S-O-M-A-L, genetics, and then we have molecular genetics. Okay, so the chromosomal genetics part is really starts off with a lot of terminology. So we have terminology of what is a chromosome? There's the chromatid. We have just organization of DNA into their structures called nucleosomes. So essentially DNA is like a string. It's a double-stranded string. So if we were to blow it up and look at it, see that there's two strands of DNA. We'll get into that in a second about structure. But those strings, to be organized, are wrapped around proteins. Called histone proteins. And for example, one clump of that's called nucleosomes. So there's a bunch of terms. There's homologous chromosome, sister chromatids. All these things you need to make sure that you're aware of, or else you won't really understand what's being discussed or the material that's being presented. So the chromosomal genetics part is about inheritance, and so that's where we get into the Mendelian genetics problems. With those, there's an assumption that for every gene, it codes for a trait. And that trait can have versions, which we call alleles. All right, so say the gene is, simple example, eye color gene. The allele is that the one gene that then codes for eye color could be blue, but there could be options. It could be brown, etc. So there's different versions of eye color gene and so forth. And so when we're talking about this, we have cells that are called 2N, which means they're diploid cells. That means for every chromosome, we actually have two of them. So two copies of the same chromosome. But the important thing to note is they're not exactly the same. One of these, say, came from your mother, and the other one came from your father. So while they have in certain locations on the chromosomes, like let's say right here is the gene for eye color, okay? And you then have two alleles for eye color. Both of those alleles could be blue, like this, or you could have both of them be brown, or you could have another option where one is brown and one is blue. So you can see, so there's these mixing up combinations. So that's the first thing. You need to kind of get the terms, and those basic ideas are lab work that we did with the worksheets. Everyone who did it should totally understand. It should be very, very easy, and all the questions about it should be very easy. If you haven't done it, then you'll be very lost and will have a hard time just figuring it out as you go along. So I recommend, if you haven't done that work, do it. Read the material about it. I have a few short lectures on this as well. Mostly on the exam, there'll be things related to monohybrid crosses. then where we actually take, say, two parents, for example, let's say from this group here, this parent could give the dominant allele, say for brown, or 50-50, the recessive allele for blue. If we mated it with another parent who also had brown eyes, who also could give 50-50 brown or blue, we'd see that their children, if they had a whole bunch of children, if they had one child, you wouldn't know, and if they even had 2 or 3 children, you still wouldn't know. But if, for example, they were able to have hundreds of children, you would see then something like this, where about a quarter of them could have blue eyes, even though the 2 parents had brown eyes, because they could have been carrying the genes for blue. They're just hidden because they're recessive. These others, they would all look brown. So 3/4 of them would look brown. So in general, that's a 3:1 ratio. That's what we call a phenotypic ratio, because that's what they look like. But the genotypic ratio is different. So you have to know that term, genotypic. That's where we actually look at the 2 alleles. So these 2 are exactly the same. So there's homozygous is the term. So again, terms, lots of terms. If you don't know what that means, you need to go back and learn, see what that means, because there's those types of things that are constantly going to come up here. So we have homozygous dominant, 1, but then we have heterozygous, the dominant and recessive combined. Okay, there's 2 of those, and then 1 that is the little b. Hopefully that's not cut off down here. So, but you basically have a different ratio for genotype. So this is a phenotypic ratio. What they look like, 3 brown to 1 blue, but genotypic is actually in this case 1 to 2 to 1, because we have 1 homozygous dominant, 2 heterozygous, but they both look dominant, and then 1 of the recessive that we'd actually see out of these 4 possibilities. So make sure you go through terms, all the terminology that's related to the Mendelian genetics. We're just looking at, really in this case, it's not a genetics course, but we want to do the basics of it. Related to the chromosomal patterns of inheritance, we also have 2 additional topics. We have the topic of mitosis, and more importantly for this part, meiosis. So it's good to study mitosis first, because that's the basic process. Meiosis includes aspects of mitosis, but it's something that's sort of different. Mitosis is for the cell division of mostly all the cells in the body. So we'll just say they're somatic cells, but we'll call them body cells. Okay. Meiosis is just for the gametes. And so these are for the germ cells, cells in your body that will give rise to sperm or egg. They undergo a special type of cell division where you go from 1 cell that's 2N, like this, ultimately through 2 divisions into 4 cells, and they're all 1N, like this. Which basically means instead of having 2 of every chromosome, they just have 1. They'll just have like 1 of these, okay, just with 1 option. So that's kind of the idea. And these might be the sperm, for example, or they could be the lead to the ova for unfertilized eggs. So meiosis is 2 divisions, division 1, division 2. We call them meiosis 1 and then meiosis 2. They repeat the same general steps, but we have some unique things that happen. Make sure you know the differences between mitosis and meiosis, and what's overall the same between mitosis and meiosis. For meiosis, the one that produces the gametes, there are 2 real key things that are very different about it, besides it dividing twice. One of those key things is that there's a point in time when you get homologous chromosomes pairing up. Does not look different enough, sorry. I'm going to redo it. So homologous chromosomes, chromosomes that would have been these kinds of chromosomes. I do them like an X here because they've doubled. So DNA replication, which will be our next topic, as we get in the molecular, DNA replication created 2 new double-stranded DNAs. Those 2 new double-stranded DNAs we call sister chromatids. All right. And those sister chromatids normally in mitosis just are what get pulled apart, separate. But in meiosis, it's different. They actually line up. They pair up with each other, and we can actually get places where they swap, 1, 2 more places, where bits of one chromosome actually end up on the other chromosome. So we actually end up getting these chromosomes like this that are hybrid. A little bit of this one goes on to that one. And ultimately, when they end up in these cells here, these gametes, we can have all these different mixtures and combinations. So the idea is, how do we get such great amount of genetic diversity? And that's because, well, one mutation can do that as well, but a lot of it is during the meiosis process, the way in which the chromosomes are separated into the gametes and the way that some of the genes can even be swapped between chromosomes creates an almost infinite amount of variation. Okay, so, but go back through the details, because like I said, I can't not go through it all. This will take hours, and my camera will run out of actually space to store this, and I won't be able to upload it anyway, and you'll be bored watching it. So I'm just trying to be quick here. So again, if you're missing something, you just need to go back to the actual lecture that explains it more. So this is just a review of what should you be studying, not exactly all the details. So the basics of genetics, mitosis and meiosis, how meiosis really connects kind of with the idea of the genetics problems, and then we get into the molecular part of it. So you need to know DNA structure. So starting off with the most basic simple unit, the structure of the nucleotide. Okay, so a nucleotide, I'll draw the carbons here, has a ring structure, which is a sugar, okay, with 5 carbons. Okay, so a lot of times it's just drawn like this. Like that. Okay, but what we're really doing, every one of those corners, there's a carbon there. Okay. Here we have an OH group sticking off it. Okay. Attached to this one, we're going to get what we call those nitrogenous bases. Okay, so we call it an N base. But what is that? That could either be adenine, guanine, thymine, or cytosine if it's DNA. And if it's RNA, it could be adenine, guanine, uracil, or cytosine. So we get one of the big differences between DNA and RNA is that DNA has one called thymine, RNA has one called uracil. In addition to that, in RNA, this particular carbon here would also have an OH in RNA, but not in DNA. DNA, the D is deoxyribose. So this sugar is a sugar called ribose. And in DNA, it's deoxy, it's missing an oxygen. In RNA, it's ribo, it's the actual ribose sugar. Okay, so differences. You'll be asked, what's the difference between DNA and RNA? You need to have a few things, 2 or 3 things that you can say specifically that's different. Okay, one is DNA has T, thymine, versus RNA has U, uracil. Second difference, RNA sugars have the OH group at the number 2 or 2' carbon, or you could say that the DNA is missing the OH group for the 2', but essentially it's the same thing. The other thing between DNA and RNA we'll get to in a second is that RNA is going to be single-stranded, but DNA will be made as single strands, but the single strands will then pair up with a second strand, so it's always going to be double-stranded, whereas RNA is going to be single-stranded. Now, attached also here to the 5' carbon is going to be a phosphate. Okay, so this is actually to be an oxygen here, and then a phosphorus. Oops, let me do it this way. Like this. Okay, so that's a nucleotide, right? Our most basic unit. You should be able to draw that, identify questions on it, answer some things. Now what's going to happen is these nucleotides get joined together when DNA is replicated. with something called a phosphodiester bond, right? That would occur between this phosphate and that OH group. These two break off, and the energy that they release when they break off is what's actually used to make this bond. Okay, and so it's going to grow, it's going to lengthen, okay, in this direction. This is the number 5 carbon. We call this the 5' end. This is the number 3 carbon, you can see here, so we call this the 3' end. If I were going to add another nucleotide here, okay, again, like this. I'm just trying to do this fast, but like I said, I go through this a little slower in the regular lectures. Same thing would happen again. Energy is released, new bond forms here. You can see it's a new 3' end. So when we talk about, we're going to talk about DNA replication. We say there's a rule. DNA replication works. It builds new DNA in a 5' to 3' direction. This is what we're talking about, and this is why that rule is that way, because we need the energy from these phosphates to create that bond. They're only broken when we add it to this side. If we try to add it the other way, it would either be upside down, which it can happen, but that's not normal. That's only in unique cases. Or there's no energy being released at all. So again, we wouldn't be able to form the new bond. So as a single strand of DNA, RNA would look the same, exactly the same, except you'd have the OHs here. That would be RNA, but overall the same thing. Know that structure. Be able to sketch it out. If you could sketch it out on your own without a reference, without anything that you're looking at, you'll do very well. Okay. If you need a reference, you will, and you understand it, you'll probably be able to do a lot of the questions, but there's some things you might miss, or you may run out of time. So if you don't want to run out of time, potentially on the exam, make sure you study this well and know exactly how to draw it on your own. The second part of this is DNA is double-stranded. So what we do is we have the phosphate attached to the sugar. Okay, that's this here. And then we have another one, right? Then there's another phosphate attached to a sugar, and there's a phosphate attached to a sugar, and a phosphate attached to a sugar, and so on. At the end of this sugar, at the 3' end, is also an OH. Sticking off of the sugars here is a nitrogenous base, right? So let's say it could be A, right? T, G, or C. Some more space. DNA is double-stranded. So I said that was one of the things I wrote up here. So we're going to have two strands come together. We're going to line them up in a very similar sort of way, okay, where we're going to get sugars lining up like this, the phosphates. But what we're going to see is that it looks a little bit different. Like this. Now, one thing I did different, you'll notice if you look at this, you say, looks just about the same, but here you put the OH on this side, 3' end, and a P on this end, 5' end. And when I did it down here, I put the OH here, 3' end, and the P over here, 5' end. They are what are called anti-parallel. Right? See, they're parallel. They run right next to each other, like this. But they go in opposite directions. This one's 5 to 3. This one's, if you go in the same direction, this one's 3 to 5. So they're different. What's going to happen is then they're going to pair together with hydrogen bonds. So this one would then have a T, an A, a C, and a G. So right there, you get the hydrogen bonds. Hydrogen bonds are what hold together the two strands of DNA, and it occurs in a very specific way. Two hydrogen bonds between A's and T's. A does not bond with G, and A does not bond with C. G and C have 3 hydrogen bonds between them. G doesn't bond with A, G doesn't bond with T, and so forth. So they're very specific in their bonding. And what that means is that as we then end here with structure, so say, okay, that's the structure. There's more to it with details about how the structure forms a helix, and there's grooves in a helix. There's major and minor grooves. Those are more advanced details. We're not going into that in this class, so don't worry about that part of the structure. Just know this part. Again, same thing. You can kind of draw that, sketch it. You're in very good shape. You'll answer any question very quickly and then move on. If you can't, then you might get stuck. So I would recommend being able to do that. Now, the next thing would be going to DNA replication. To replicate DNA, it's exactly what I just did here, essentially. What happens in the cell is that the chromosomes are made up of DNA. The DNA is double-stranded. Okay? And before mitosis, or meiosis is going to occur, this double-stranded DNA structure has to be doubled. We have to now have 2 of the chromosomes so that we can then separate them into new cells, and the new cells are exactly the same as one another. So what's going to happen is there's going to be a site called an origin of replication. It's going to be a place where the 2 strands of DNA are going to start to become pulled apart. Right? We're going to actually start to pull them and separate them by breaking some of the hydrogen bonds between the 2 strands. And so they get pulled up this way. Remember, the one that says A, T, G, C here, and this one has the other complement. We call it the complementary strand. It's not the same sequence. It's not A for both of them, T for both of them. One is A, one is T. So they complement each other. They're not exactly the same. We pull that apart. There's an enzyme that does that. So I don't have the time to go through all of them. You have to go back and look at the lecture. So helicase pulls them apart. Single-strand binding proteins keep them apart. So the helicase will start working and pushing them apart in this way. Other enzymes will come in. A primase will actually start building little pieces of RNA along here so that a new enzyme called DNA polymerase will be able to come in, and it will actually start to be able to make the new strand. A couple things about DNA polymerase you need to know. DNA polymerase is the enzyme that makes the new strand of DNA. So it's the one that actually puts together the nucleotides, breaks the phosphates off, makes the phosphodiester bond, makes the strand longer and longer and longer. It's using the other strand as the template. So it's saying, if this one here has an A up here, what nucleotide should I put here? Should it be another A? Should it be a T? Should it be a G? Should it be a C? There's only one option. T. A and T go together. That's the only one. So DNA polymerase will put that nucleotide in that place. It will make that bond, and it will make this particular strand. It'll make a strand grow. It has rules. It's template. So it reads the template, and this is very important. There'll be probably several questions, okay, on this type of thing. Reads the template in a 3' to 5' direction. Makes new DNA 5' to 3'. Remember, they're anti-parallel, right? 3, 5, 5, 3. The one strand has to be opposite the other strand. So the new strand that's being made is running the opposite direction of the one that's being used as a template. It has to make the DNA in this direction, 5 to 3. Remember, because this is the 3' end, and if we're going to add the new one here, like that, to make this bond, that means we're going to have a new 3' end, so it's going to be growing in the 5 to 3 direction. Okay, so those are a couple of rules. You have to make sure you know those rules, or else it'll be all very hard. The little things called the primers get removed later by proofreading. They get replaced with DNA. There's an enzyme called ligase. It comes in, it seals them up. There's something else totally different that's working at another location called topoisomerase that's actually, relieving the coiling. The strands are twisted. It's actually making them a little more ladder-like. Normally they're actually all twisted like this. So again, those are all other material. There's details that's specific to this exam that you should know and be familiar with. So you need to go to the lectures if you don't know those, and read sections of the book that talk about them. And there's other accessory videos outside of what I have put in there through Khan Academy and other places that go over it. So go over it as many times as you need from as many sources as you need until you get it, till you understand what's going on. So that's DNA replication. Now, that's part of it. So that's part of this material. after we go over DNA structure, then that structure leads to DNA replication. So we got structure, we got replication. Then we're going to start to have what we call gene expression. And gene expression is broken into two processes. There's transcription and translation. And people often confuse them. So what you have to know is that what's going to happen is one of these strands of DNA is going to serve as a template for a piece of RNA to come in. Okay, so a different enzyme called, not DNA polymerase, but RNA polymerase is going to read one strand of DNA. Okay, so one of these two. It's not going to read them both. It's just going to read one. It's just going to read one of them. And what it's going to do is it's going to then make a piece of RNA. So it's going to do the same kind of thing where it would come in here, sugar, with a C goes a G, with a G goes a C, with a T goes an A, with an A goes a U. Okay, because now this is a piece of RNA that's being made. So all these are RNA nucleotides, a little bit different than the DNA. Very much the same, almost exactly the same, but different. Okay. Another difference is when they bond with each other, it's very temporary. And once they're done, this piece leaves. Okay, so our GCAU piece of RNA that we just made will separate from the DNA. That's the transcription process. Using a DNA template to make a piece of RNA. The DNA, what will happen here is it'll pull apart, or it'll be pulled apart so that that can occur, but once the process is done, it'll just go back together again. It's not permanently pulled apart. It's not broken or damaged, nothing like that happens to it. It just pulls apart. One strand's used, then it goes back together. It makes a copy, essentially, of one strand. That's what's going on. Transcription, to transcribe means to copy. We're making a copy of one of the strands. That's it. This piece of RNA could be one of several types of RNA. So the next thing you need to know is after transcription is transcription makes RNA. There are different kinds of RNA. You have to go through the processing part of it. There's messenger RNA that actually carries the code for a protein, polypeptide, that's going to be made. There's transfer RNA, who's going to carry the individual amino acids, who will be put together to make that protein. And then there's the ribosomal RNA, who is essentially the little machine, the enzyme, who's going to read the information from the messenger RNA and take the amino acids from the transfer RNA and put them together. So the ribosomal RNA is one who actually will make the polypeptide. So this one has the code for the new protein. This one has the amino acids for the new protein, and this one puts it together. So the product of translation is going to be a polypeptide. So what you'll need to do is also go through that process. That process has a whole number of steps to it. Initiation, elongation, and then termination. Know there's some rules about start codons and stop codons. So the code is in units called codons. Make sure you know that. There's a codon table. I'm telling you right now on your exam, I'm giving you a codon table. It's there with the written part of the exam. You'll be able to use it to decode information. So kind of take that. You can use other codon tables than the one I gave you. They should all have the same information. The one I gave you I know is correct though. So you'll have the quick overview of the process. Messenger RNA contains the code. You're going to get the ribosomal RNA first, a tiny subunit, the small subunit they call it, small subunit ribosomal RNA. binds to the messenger RNA with a transfer RNA attached to it. And that transfer RNA carries a single amino acid. It'll read until it finds the code, the very specific code that codes for that amino acid. And once it gets there, then the large subunit will come in. All right, and that's the part that we call initiation. Or actually, all this part is called initiation. Once we get to that point, right, then new transfer RNAs with amino acids will start entering this ribosome. And then the ribosome will make a peptide bond between the two of them, and it will grow that string, that chain of amino acids, and then it'll move, and it'll just keep moving along the messenger RNA, reading it. Difference, a little bit different here. It reads messenger RNA in a 5' to 3' direction. So the DNA polymerase reads DNA in a 3 to 5 direction, but a ribosome reads messenger RNA 3 to 5, the other direction. And those directions are important to know. It seems like it's a small trivia specific detail, but it's actually very important to explaining the structure, the underlying structure of these molecules, explains why they work the way they do, explains why the process happens the way it does. So if you understand them, the structure, then this will make a lot more sense. Eventually it reads a codon, a code that says stop. Okay, there'll be one that says stop. UAA, UAG, UGA. Those are 3 codes that say stop here, and then it stops, and then you end up with a polypeptide. So you end up with a polypeptide made, a string of amino acids all put together. That's kind of the end. That's gene expression. So that may not be functional. So usually it goes on to the endoplasmic reticulum and the Golgi apparatus, and that's where it's processed into a functional protein. So the whole point of gene expression is to get DNA genes who do nothing. DNA does nothing. The genes do nothing. But it's to use the information in the DNA to make a protein that has a function for the cell. To do that, we have an intermediate part which involves the RNA. So the RNA is kind of the go-between. Okay, it's actually the one who does all the stuff, carries the code, translates the code, carries the amino acids who are going to make the polypeptide or protein. It does all the work, really. Then the protein carries out a unique job in the cell. So that protein could be your sodium potassium pump. It could be a receptor. It could be a G protein. It could be all these other molecules that have jobs, membrane proteins, enzymes in glycolysis, all those sorts of things could be who that protein is, who actually do all these things for the cell. So this is kind of a little longer than I thought, because that's just the 4th exam material, the 100 points. There's also cumulative, and I'm just going to go very fast on the cumulative stuff because I'm going to keep it specific, try to tie it down to something so you don't have to study every single thing that was on the previous exams. You could just kind of really focus on just a few topics. Okay. So roughly, not exactly, but it's going to be roughly 50 additional points on your exam. So 100 points from all this stuff, 50 more points cumulative. Okay, that's going to be exam 1 material, which is the chemistry. Things to emphasize from that had to do with bonds, polar, nonpolar, which had to do with electronegativity, You can see the hydrogen bonds still come in now, later on, in DNA, holding the DNA together, all these things. So if you didn't understand the chemistry in the beginning, a lot of these things later on are just kind of foreign, the terms or have less meaning to them. They have more meaning if you understood all the underlying chemistry and processes. So make sure you focus on water. Water molecule, because it's so important for cells, has an oxygen. It needs 2 electrons, so it can form 2 bonds. Hydrogens have 1 electron. They only form 1 bond. 1 shell, 2 shells. Review that chemistry. If it's not familiar to you, if you don't know what I'm saying right now, you need to go back then and study more. Oxygen then is unequally sharing the electrons, though, right, with the hydrogen, so it becomes partially negatively charged. These left with their protons feel partially positively charged. That makes this not only are these polar bonds, that creates a polar molecule, right? That means molecules of water will interact with each other through hydrogen bonds, like this. Molecules of water will interact with other polar things. So anything else that has oxygens that carry the partial negative charge, the hydrogens will then attract to. Anything that has positive charges, the oxygens will attract to. So anything sugars, any proteins, the DNA, the RNA, all these molecules have charges and that have polar parts to them. They're all going to interact with water in a very specific way, as will the phospholipids that make up the cell membrane. So part of this was then phospholipids. So we go to the second part, second exam 2, which was membranes. Make sure you know membrane structure. So make sure you know what is a phospholipid, how are phospholipids oriented in the presence of water. The heads like the water, but the tails don't. Remember, the tails are non-polar. The tails have no charge, no partial charge. They don't like water. They move away from water. Water moves away from them, but the tails move toward each other. So we have our non-polar tails, and that creates a bilayer, a membrane, that then gets populated with other kinds of molecules. So be able to sketch out a whole membrane, explain characteristics of the membrane. There are proteins then that associate with the membrane, transmembrane ones, peripheral ones, generically. You can call that G protein, but G protein is just a certain kind of peripheral protein. If I ask generically membrane structure, you don't say G protein, you just say a peripheral protein. Because there's tons of different kinds of peripheral proteins. G proteins aren't the only ones, but you might be familiar with those. The sugars that attach just to the outside of the cell. So the sugars might attach directly to the phospholipids. The sugars might attach to the proteins. Sugar protein, glycoprotein. Sugar lipid, glycolipid. Okay? And then we also have cholesterol that's stuck in there. So a little bit about the fluidity the cholesterol establishes and buffers for our membrane. So we have phospholipids and cholesterol. We have transmembrane and peripheral proteins, and the glycolipids, glycoproteins. Okay, so make sure you kind of do that. Overall, diffusion then is when things are going to move across the membrane from one side to the other, but the membrane's a barrier to many things. Anything that's big, anything that's polar, anything that's charged, and the things can't get across. So we have to have our channels, pores, active transport pumps, something that's going to be able to move molecules or ions across a membrane. So something binds here depending on its concentration. So whatever the concentration of this thing is on this side versus this side of the membrane will determine whether that process is going to be diffusion or active transport. So it's the kind of thing you need to be able to interpret. Okay? If this is high and that's low, this process would be diffusion, right? High to low is diffusion. Energy be given off. If it was the other way around, right, and this is going to be moving from low concentration to high concentration, this process here is going to require energy, right? So it's not diffusion, right? It's going to be active transport. And if it's active transport, then we have to couple it with something else, with energy. So we're usually going to need ATP other things can be used. But we're often going to need ATP to give off energy to do that. Okay, so make sure you kind of know how that part works. That does bring us to the third exam and the last material, and it ties into this. Hopefully you'll see the main things I chose for topics that are the cumulative ones tie together in many ways with each other, all right, as well. ATP. Cells need ATP then to do all the things that they do. So the third exam on enzymes and metabolism is really about how does a cell make ATP, okay? So you have sugars like glucose, which eventually get oxidized, all right, which means electrons are removed from it, okay? The electrons are typically removed from the hydrogens, and so we really get carbon dioxide and something called NADH made. The NADH is what's carrying the electrons. So the energy from the sugar really goes to NADH. The carbon dioxide is waste. Okay? What does the NADH do? The NADH goes to the mitochondria in the cell. And in the mitochondria in the cell, NADH delivers these electrons to proteins who are active transport pumps. So what they're going to do is then they're going to use the energy not from ATP, but they're going to use the energy from electrons. And they're going to pump protons across a membrane. It's an electron transport chain, so there's not just one, but several proteins. And the electrons will go between all these proteins, ultimately toward oxygen. They all work to pump protons across a membrane, which then makes the proton concentration high, okay, on this side of the membrane, which means it wants to come back across the membrane, and when it does, that means that the protons move by diffusion. Okay, so if these were the protons going across the membrane, that gives off energy. And in this case, instead of using ATP, this process is one that'll take ADP and phosphates, put them together, use the energy from the diffusion to make ATP. So big bulk of material that's on your third exam was about how the cells make ATP. Where does ATP come from? The whole metabolism, glycolysis, pyruvate oxidation, citric acid cycle, electron transport chain, FO, F1, ATPase, all that stuff really combined could be summarized by sugars and other nutrient molecules lose electrons, oxidation. They go to NADH. NADH delivers them to electron transport chain, who creates a concentration gradient and uses diffusion to make ATP. Okay, that's super, super simple, quick summary, but that's kind of the idea. So there'll be a couple questions about probably electron transport chain, in particular the NADH part and the oxidation part. So don't worry about details of any of the processes specifically. Look at the overview, their general equation for what goes on there, but don't worry about all the reactions and everything of them, mostly this part of it. So you'll have a couple questions in the multiple choice on these, and there'll be a couple of your essay-ish questions related to these three main cumulative topics I brought up. So there's a lot of material, but the majority of it, the overwhelming majority, is going to be focused on the DNA part of it and the chromosome part of it. And then there'll be some stuff that you've seen before that actually, and much of it ties into this as well. Or there's some aspect of it. Okay. So that should be it. If you have questions, let me know. The exam is available on Monday. Real early, just right after it turns Monday, from Sunday to Monday, it's like 1 a.m. You'll just see it there, both the written and the online part. For the online part, once you start it, you have 60 minutes. You have one chance to do it. That's it. Make sure you're well prepared to do it. The written part, you just print off and we'll answer them anytime through the week. You'll read the instructions. You could choose some and all. It all has to be done by Friday, 11 p.m. So you have the whole entire week to work on these and or do them, do the work, do the written stuff. So make sure you put aside time to do it. Make sure you're prepared and ready to go.